So we're going to be looking at two other elements on chromosomes which are required for their stability and maintenance. We looked last time at regions of DNA replication. Of course, those are required for replicating the genetic material, and of course stability of the genome and maintenance of the genome. And so we're going to look at two additional regions. The first are telomeres. And telomeres are required for proper replication of the ends of the chromosome. Turns out there is an issue at the end, and I'll show you some diagrams to help you understand what's going on. And then, of course, centromeres are required for pulling the chromosomes apart during mitosis, and if those centromeres are defective or not working, then the two chromosomes don't split apart. Okay, so let's start looking at the telomeres first. So, without a special mechanism to replicate the ends of the linear chromosomes, DNA would be lost during each round of cell division. Let me explain what's happening. Also, I will also say they're required for protection of the ends of the chromosome because there's nucleases in the cell that could degrade the ends of the DNA. But most important is the ability to replicate the ends. So let's look at what happens here. So look at the very top. You have what we've seen before, obviously, 5' to 3', the orange on top. On the bottom, there's the anti-parallel. On the lower strand, you have the continuous DNA synthesis. The 3' hydroxyl is represented by the arrow. And then on the upper strand, you have the lagging DNA synthesis, where you have the RNA primer made, DNA polymerase, another RNA primer. Those are the Okazaki fragments. And of course, eventually the RNA gets removed and the DNA fills in, you get ligation. But as the fork opens and it reaches the end of the chromosome, look and see what happens down in the middle. It says replication fork reaches the end of the chromosome, and let's see what happens. On the leading strand, the synthesis continues to the very end. On the lagging strand, you have these RNA primers, and that first RNA primer, or I should say the last RNA primer, the one on the most right-hand side, that is going to get removed. It's got to get removed by the FEN1 and the RNase H complex, or it's going to get removed because RNA is inherently unstable. It's not going to last for a long time. So you look down at the bottom, and what you see is that RNA primer is gone. The other Okazaki fragments have ligated together, but that RNA primer on the very right-hand side is gone. Now, that cannot be replaced by DNA. It cannot be replaced by DNA because DNA requires an already existing 3' hydroxyl to extend. So the DNA polymerase cannot sit down in that DNA and make and use the template and make DNA. So that information is lost. So since that represents around 10 to 15 nucleotides at the end, because that's the size of the primer, around 10 to 15 nucleotides, and we multiply that by all the ends of all the 46 chromosomes, we would end up losing around 1,000 base pairs per cell generation, per cell division. And of course, we don't want to lose information, and so there has to be a way to completely replicate that. So this is referred to as the end replication problem. In a linear DNA, you cannot replicate the end of the lagging strand. So we have this end replication problem. And that is the DNA polymerase cannot replicate the 3' end of the linear chromosome because it needs a primer, it needs some pre-existing 3' hydroxyl to extend. And that's not there, so the RNA is removed and you have that missing information. So the end replication problem, turns out there are 4 known ways that this end replication problem can be solved. The first is to have an RNA genome. And I mentioned that there's many viruses that have RNA genomes. And so with an RNA genome, which is replicated by an RNA polymerase, there's not a problem replicating the end, even on a linear chromosome, because the RNA polymerase does not require an existing 3' hydroxyl to extend. It can just sit on the template and start making RNA. So that's one solution. You have an RNA genome. Of course, it's not a great solution because RNA is not stable, but still that's a solution. The second is to have a circular genome. If you have a circular genome, there's no end. So if you can imagine a circular DNA with a single site of replication, the replication goes around that circle, and then it just finally reaches the end, and it's right in juxtaposition to the initiating nucleotide, and then you can have a ligase that can connect those. So a circular genome is another solution, and of course many bacteria have circular genomes. Another solution which has been discovered is what's called by using a protein primer. There's a number of DNA viruses with linear DNA genomes that have this kind of protein primer, and what happens is there's a protein called a cap protein that binds to the 3' end of the strand of DNA. And that's what's shown. The straight line there, it should say 5' on one side, 3' on the other side. And then what has to happen in order to replicate that, there has to be some 3' hydroxyl for the polymerase to extend. And so there's this cap protein that has a tyrosine. Tyrosines have an OH group, hydroxyl, as part of the R group. And so the three-dimensional structure of this cap protein is such that it binds to the end of the DNA, and it positions the positions the hydroxyl of the tyrosine in just the precise location that allows the polymerase to bind on to that OH of the tyrosine and use it as a primer. So it will add a nucleotide to that, and then it will extend, the synthesis can extend. So the protein, in a sense, primes the synthesis. And finally, what happens in our cells, in mammalian cells, is that there are telomeres. Telomeres are 6 base pair repeats, and you can see those at the bottom of the drawing at the bottom. There are 6 base pair repeats. In humans, the repeat is GGGTTA, so it's repeated over and over and over and over again. These telomeres are added to the 3' hydroxyl, the end of the strand. And that's what's represented by those little arrowheads. So each arrowhead represents another telomere repeat. And the ends of chromosomes can have 100, 200, 500 repeats. So they're long telomeres. These are the telomere structures. So what happens is DNA synthesis will occur on these telomeres. So you'll have the RNA primer made, and that's shown by that squiggly line. DNA synthesis will then extend from that primer. And when that primer is removed, you're still losing information, but all the only information you're losing is copies of the 6 base pair repeat. So there's no genetic coding information. There's no information there. So it acts like a buffer in a sense. So information is still lost, but all you're losing are these repeats which have no function other than to act as a buffer. And then in any case, at the next round of DNA synthesis, more repeats are added. So it's a solution. The problem's still there because you still lose that primer, but you're not losing any. You never get into the chromosome per se. So the enzyme that adds these repeats, adds these 6 pair repeats, is called telomerase. So here we look at what telomerase does. So as it says, telomeres and the telomerase in particular, because they add repeats at every round of cell generation, every round of cell division, they prevent the linear chromosomes from shortening because of this problem. So let's kind of walk through this. So first of all, on the right-hand side, I have the repeat sequence, GGGTTA. That's in human. Different organisms have different repeat sequences, but ours is this. And so if you look at the very top, we're going to look and see how the polymerase, this telomerase, acts. So if you look at the very top, you see the issue. You have the DNA synthesis, which is occurring. The red represents the newly synthesized DNA or RNA primer. And so you can see that information is lost. It's showing telomere repeats. But at every round of DNA synthesis, the telomeres are extended outwards, and the telomeres represent those orange repeats. And they're extended by telomerase. Telomerase binds to the upper strand of DNA at the 3' end. It's a large multi-protein complex, and what happens is this will synthesize DNA repeat, GGGTTA. Now, the telomerase is a DNA polymerase because the telomeres are DNA. They're these DNA repeats. And what I told you when we talked about DNA polymerases is that every DNA polymerase requires a 3' hydroxyl, which is obviously there on the DNA, but it also requires a template in order to know what to put down. Now, there's no template DNA available, so what the telomerase does is it's pretty amazing. It carries within it a short strand of RNA, which acts as the template for the repeat telomere sequence. And that's what's shown in blue. So the telomerase is bound, has its RNA template, and the template gives the instructions of what DNA to synthesize. So the RNA template I've also shown on the right-hand side. The RNA template is a short stretch of DNA, which contains the sequence CCCAAU. That's the complement. You can see the complement to the repeat sequence, but of course it's RNA, so it has U. So it knows exactly what to synthesize. Now, two things to notice about the template. First of all, of course, it's in the anti-parallel orientation. So when it synthesizes, it will synthesize GGGTTA. But in addition, there are two additional nucleotides that are not part of the template per se. They're not synthesized into the repeat, and that is AU. That's the AU at the very beginning. And what that AU serves to do is it serves as a marker, so to speak, in order to move the telomerase down the line and line up appropriately to make the next telomere, because that AU will base pair with the TA at the 3' end of the last telomere. So the telomerase makes an RNA makes the telomere sequence, telomere repeat, and then it moves down, base pairs with the TA, and then uses the rest as a template to make the GGGTTA, then moves again down, base pairs with the TA, and keeps repeating that, does it over and over and over and over again. So it makes these telomere repeat sequences, and then of course if you look on the very bottom, what can happen is then you have this buffer region that even if you lose the RNA, all you're doing is you're just losing a little bit of repetitive DNA, which has no coding sequence, no information. Now, at the very beginning of the semester, not the beginning of the semester, but a couple of lectures in, we talked about cell division and the fact that normal human cells cannot grow indefinitely in culture, and that was the Hayflick limit. They had about 50 cell generations for normal cells, but on the other hand, cancer cells and stem cells were able to grow indefinitely. And at that time, what I told you was that there's probably some kind of an internal clock or internal counting mechanism that can evaluate the number of the number of cell divisions that the cell can undergo, and what was responsible for that was the telomeres. That the telomeres somehow count the number of cell generations, and it turns out that's what happens. Now, let me just give you some of the information that we have. What we know is that cells with limited lifespan, like cells that will undergo the Hayflick limit at 50 cell generations, what we see is that as the cells grow, the telomerase activity decreases. Telomerase activity just begins to decrease, and the telomeres shorten. The telomeres shorten. Now, in stem cells and in cancer cells, the telomerase does not decrease its activity. It stays very high. And the telomeres are stable. They remain very long. And we also know, and I have this written down on the slide there, that the telomere repeats are bound by proteins. There's a protein called TRF1, a protein called TRF2, there's a protein called RAP1. There's a series of telomere binding proteins that bind onto these repeats. And so what happens is when the telomeres shorten, these repeats become unavailable for binding of these proteins. Now, so if you look at the graph below, what you can see is that this is kind of what I showed you earlier with the Hayflick limit. What you have is the cells are growing. That's the solid line. Cells are growing. When they reach the 50 cell generations, they go down and they die. So that's the limit of cell division. Now, if we add a tumor virus to these cells before they have a chance to completely die, or sometimes there's compounds that we can add that can mutate the cells. But if we add a cancer-causing virus or a tumor virus to these cells, the cells will become tumorigenic and they'll start to grow again. That's what's shown by that line going up. Now, if we look at the telomerase or we look at the telomere length, that's shown by the dotted lines. And so what we see is that the telomerase is active, the telomeres are a substantial length, but as the cells divide and they reach that Hayflick limit, the telomerase is decreased and telomere length is shortened significantly. So there's this clear correlation between the activity of the telomerase, the length of the telomeres, and cell division. Now there's two possibilities to explain this kind of correlation. One possible explanation is that the telomeres shorten and telomerase activity decreases because the cells are reaching the Hayflick limit. That is, the cause of the telomere repeat decrease and the cause of the telomerase decrease is because the cells are beginning to die. The other possibility is that the cells are beginning to die and they lose their proliferative potential because the telomeres are shortening and the telomerase activity is decreasing. So that would mean it's a cause of the Hayflick limit. And there's experiments that have been done that have shown the second, that is, the loss of telomerase activity and the shortening of the telomeres is a cause of the Hayflick limit. And we know that from the following experiment, and that is people have been able to take the telomerase gene or telomerase protein and inject it into cells that are undergoing the Hayflick limit. So and what happens is the cells start to grow. The telomeres increase and the cells start to grow. So what that says is that all you need to do is add telomerase to these cells, and you will increase the telomere length, and you will bypass the Hayflick limit. Now, one explanation is why is it that shortening the telomeres leads to a decrease in cell division? What's the connection there? And it could have to do with the proteins that I mentioned are bound to the repeats, the TRF1, TRF2, and RAP1, and some other proteins, that they may have multiple functions. So that the binding of these proteins to the telomere repeat occurs, but as the telomeres shorten, these proteins are released because the binding sites disappear. So these proteins are released, and there's some suggestion that some of these proteins may have a negative regulatory effect on DNA synthesis. So when they come off the telomere, they feed back onto the replosome and somehow cause it to begin to stop. So that's one explanation for the role of the telomeres in counting cell divisions. Now, we've looked at many cancer cells, and the telomerase is very active. And so people have thought, well, maybe this could be used as a treatment for cancer. That is, if the telomerase is responsible for the long or the continual cell growth, the continual proliferation, then if we inhibit telomerase, then we should be able to eventually get these cells to stop growing, and can it be used as a tumor therapy? And it turns out that it is being used as a tumor therapy. This is something that's very new. I'll show you. There's a company called Geron. This Geron Corporation, and the goal of this company is they focus primarily on telomeres and telomerase, and what they've been working very hard to do is to come up with cancer therapies based on the inhibition of telomerase. And in fact, they have come up with some drugs, and here they have one that they're showing, imetelstat, which is, they're calling a first in class. That means the first generation of these telomerase inhibitors, a unique model for targeting cancer. And so there are some successes with this. And here it shows, I've taken these from papers, a couple papers that have been published. The first one shows that this particular inhibitor, telomerase inhibitor that they have come up with, this was published in 2016, and I guess by now there's more success, but they say it does have some activity across non-small cell lung carcinomas, that there is some potential use for that. And then in the lower one, in Leukemia, Journal of Leukemia in 2017, again, this same telomerase inhibitor has differential effects on normal and malignant cells. So they are clearly using this to move ahead for some kind of a cancer therapy. So that is telomerase and the role in the replication of the ends of chromosomes. And I want to finish up this little section by talking about centromeres, which was the third element required for stability. We're not going to spend that much time on them. I'll just tell you a little bit about them. So the centromeres are required for proper partitioning of the cells during mitosis, particularly. And centromeres occur on each chromosome. They can be in slightly different positions, and they are required, as it shows in this diagram, to pull the chromosomes apart during replication. So you have during S phase, you have the synthesis of new DNA, and that new chromosome, those chromosomes are held together at the centromere, and then microtubules bind to the centromere and then pull those apart. And so if you're missing a centromere, or if there's a defect in the centromere, then the chromosomes don't get pulled apart, and you end up getting two of the same chromosome in one cell, and that gives rise to example for something like trisomy 21 or Down syndrome, where somebody would have 3 copies of chromosome 21 due to defective centromeres. So how do these things work? So the centromere is attached to microtubules. So if you look at the left-hand side, it shows the replicated chromosome. It shows where the centromere is. It's in red. And by the way, centromeres are DNA sequences. They have very specific sequences. So I have that typed out underneath the title. It said the centromere. Then I have a series of sequences. So there's a sequence GTC, ACA, TGA. And then there's a long region of 85 base pairs, which are primarily T's and A's, AT rich. And then there's another sequence. So it's around 100 base pair sequence is required for the centromere. This region binds to proteins. So the sequence represents a sequence that proteins bind to. So if you look at the left-hand side, you see that in the red they call it kinetochore, and then microtubules bind to the kinetochore. And it's thought that there's around 30 to 40 microtubules bind to each kinetochore. The kinetochore are a series of proteins. This diagram doesn't really show it the way I would like to show it, but the kinetochore are a series of what are called centromere binding proteins. And there's many different proteins that are complex onto this cluster that bind to that specific DNA sequence. And then to that DNA sequence, to that kinetochore binds an additional cluster of proteins, which are called basically connecting proteins, not very creative, but that's a connective protein complex. So there's proteins bound to the DNA, kinetochore. Then there's the additional connector proteins that bind to the kinetochore, and then those proteins bind to the end of the microtubule. And what's shown here is only one, but it would be a longer region. There may be 40 to 50 of these bound to microtubules. Now the chromosomes then are pulled by 2 different mechanisms. One mechanism is that the microtubules shorten, and as they shorten, the chromosomes get pulled. But there's another mechanism, and that is some of the connecting proteins, in fact some of the proteins in the kinetochore, have motor activity, just like kinesins or dyneins. So the motor proteins are activated, and they basically move down the microtubule and pull the chromosomes apart. So it's like a dynein-like movement that pulls these chromosomes. Okay, so that's it. So we looked at DNA replication, we looked at the telomeres, and now we've looked at the centromeres, all important and required for what we call protein or chromosome maintenance or chromosome stability.